We'll start again this afternoon with the passage out of the Bible that supports what we're doing this afternoon. We have it in a library because it's good to have it in a place that's not a church building. The reason for that is it's more likely that folks will come visit with us who may not be believers. And so if there are any of you in the audience who are in that position, thank you for being here. And it is also for those of us who are believers in God to further support that faith. And I hope that you will leave this last session with a deeper appreciation for the amazing God that we serve. Romans, the first chapter, chapter 1 and verse 20 says, "For since the creation of the world, his invisible attributes are clearly seen, being understood by the things that are made, even his eternal power and Godhead, so that they are without excuse." So the Bible teaches that nature itself can convince a reasonable person that there is a God, and that God is eternally powerful, and he's separate from nature. He's divine. Let me add again, science cannot investigate the supernatural. You hear what I said? Science can only deal with nature. And some people say because of that, science says there's no God. That's exactly wrong. No, science supports evidence that points to a designer. And if there's a designer, he has to be outside of this nature. So you can use science to direct you to a belief in God. And in fact, that's what should happen. But you will never prove it absolutely. And science cannot test a supernatural being. It's impossible. Science can only deal with nature. But God himself has put nature here. And in every generation of mankind, he's provided enough evidence in nature to convince any reasonable person there's a God. And I'm here to tell you that in this generation, he's provided more evidence than ever. And I'm convinced it's because young people who are coming along now need more evidence because they're being bombarded with negativeness about God. So he's opened the floodgates. So welcome to Opening the Floodgates, class. I want to overwhelm you with the amazingness of the God we serve. As we said this morning, I want you walking out of here stupefied with the complexity of what our God has made. And this passage from the Bible supports what we're doing. And hope no one takes offense that for the rest of our time, I'm closing this book and turning to God's other book, because he does have another book. And all the famous scientists of the 16 and 1700s would tell you we're thinking God's thoughts after him. And so that's what we're doing today as well. All right. Welcome back, class. You are my class. I don't allow sleeping. I expect rapt attention. And if after lunch, you had a nice big lunch, you start fading out on me, we're going to do something about that. Because if I were sitting in your place, I'd be falling asleep. I had a nice lunch and, that's nap time. But we don't allow it. So, I have mentioned this to you in our previous lecture. Had you been in school, in college, in England or in Europe in the 1800s, every college student takes a course like this. It's called Natural Theology, or the evidences of the existence and attributes of the deity collected from the appearances of nature. This particular one happens to be by William Paley. It was one of the most famous books of that time and was used widely. Charles Darwin studied William Paley's Natural Theology in college and was deeply impacted by it and wanted to be a preacher. Did you know that? Charles Darwin was training to be a preacher until he took that trip around the world on the HMS Beagle, which caused him to begin thinking otherwise. And what he began thinking was the old notion that seems to be in the Bible that every species was created as it is, where it is, didn't look like that was the way it was. And he began changing his mind about that, and eventually wrote the famous book on the origin of species, in which he took the position that you can get new species without God. Now I'm pausing there a minute, because I want you to understand that species is not in the Bible. Okay, can I get biblical with you here just a minute? The word species is a man-made construct. So what is a species? Generally, it's something that doesn't interbreed with something else. That's generally how we define it. But it's hard to define species exactly, class. And whether or not God created every species the way it is and where it is not the issue, because when you look in Genesis 1, it says they were created and then they reproduced after their what, class? After their kind. So what's a kind? That's hard to demonstrate from Scripture, exactly what that is scientifically. The Bible's not a science book. Well, is there some variation enough that you may get a new species because that's not what God created, because the kind has variability in it? I think that's possible. But I'll tell you what the Bible clearly teaches. There's a line beyond which you're not going to change anything. The Bible calls it kind. Darwin had this class. And this class is also referenced by Richard Dawkins, our favorite modern critic of God. So what I thought I would do this afternoon, I have both of these books. Here is kind of an upgraded William Paley's Natural Theology. If you're studying some of this with your children, this would be a good book to use, because William Paley does a great job with this in his time. But I want to show you what Richard Dawkins does with that. So I start with Richard Dawkins' book, The Blind Watchmaker. And I'm going to explain to you where he's coming from. So I'm on page 1, and here's how he starts his book. We animals, that give you a clue? We animals are the most complicated things in the known universe. Would you say amen to that? I think so. Then he goes on to say, biology is the study of complicated things that give the appearance of being designed. That's how he starts his book. And then he spends 300 pages trying to convince you that they were not designed. I'm telling you the truth. That's how he starts. They give the appearance of being designed. Let's go on. This is on page 3. The process by which an airliner came into existence is not fundamentally mysterious to us because humans built it. Agreed, everybody? The systematic putting together of parts to a purposeful design is something we know and understand, for we have experienced it firsthand even in our childhood. You kids ever build erector sets? Yeah, you did it with your brain. So we know how to build things and how that works. So then he says, what about our bodies? Each one of us is a machine, like an airliner, only much more complicated. Were we designed on a drawing board too? And were our parts assembled by a skilled engineer? It's a surprising answer, but the answer is no. We have known and understood it only for a century or so, when Charles Darwin first explained it. This man is a Darwinist to the depths of his being. He's the ultimate reductionist. So let me go on. The watchmaker of my title is borrowed from a famous treatise by the 18th century theologian William Paley, Natural Theology. Published in 1802, it is the best-known exposition of the argument from design. Class, have I been saying that from day one? Argument from design. That's what Romans 1's talking about. Well, he says Paley was the best one. It's always been the most influential of the arguments for the existence of God. It is a book I greatly admire, he says, for in his own time its author succeeded in doing what I am struggling to do now, says Dr. Dawkins. He had a point to make, he passionately believed in it, and he spared no effort to ram it home. Have I been ramming stuff home? And I make no apology for that, folks, because Dawkins is full of ramming stuff home. And he passionately believes it. Then he goes on to say he had a proper reverence for the complexity of the living world, and he saw that it demands a very special kind of explanation. The only thing he got wrong, admittedly quite a big thing, was the explanation. He gave the traditional religious answer to the riddle, but he articulated it more clearly and convincingly than anyone had before. The true explanation is utterly different, and it had to wait for one of the most revolutionary thinkers of all time, Charles Darwin. Paley's argument, made with passionate sincerity and informed by the best biological scholarship of his day, is wrong. Utterly and gloriously wrong. Well, this book starts, the book by Paley starts with the famous passage. May I read it to you in British? In crossing a heath, suppose I pitched my foot against a stone, and it were asked how that stone came to be there. I might possibly answer that for anything I knew to the contrary, it had lain there forever. Nor would it perhaps have been very easy to show the absurdity of this answer. But suppose I had found a watch upon the ground, and it should be inquired how the watch happened to be in that place. I should hardly think of the answer which I had before given, that for anything I knew, the watch might have always been there. No, the watch must have had a maker. That is, there must have existed at some time and at some place or other, an artificer who formed it for a purpose which we find it actually to answer, who comprehended its construction and designed its use. Whereupon, class, he spent the rest of his semester showing you nature is much more complicated than a watch, by far. Here's what Dawkins says. The analogy between a telescope and an eye, between a watch and a living organism, is false. All appearances to the contrary, the only watchmaker in nature is the blind forces of physics, albeit deployed in a very special way. A true watchmaker has foresight. He designs his cogs and springs and plans their interconnections with a future purpose in his mind's eye. Natural selection, the blind, unconscious, automatic process which Darwin discovered, is the explanation for the existence and apparent purposeful form of all life. But it had no purpose. It has no mind, it has no mind's eye, it does not plan for the future, it has no vision, no foresight, no sight at all. If it is to be said to play the role of a watchmaker in nature, it is the blind watchmaker. So can I summarize all of that? Natural selection, he says, is the blind watchmaker that can produce everything you see, every living thing. He assigns it the very property of God itself. And he spends 300 pages trying to convince you natural selection can do this. You don't need anything else. So part of my job tonight, or this afternoon, is to help you see where in the world these guys come from with this. It's just almost unbelievable, isn't it? He believes it with all his heart. So much so that he will not have you teaching science to anybody if you don't believe that. So I'm on his blacklist. All appearances to the contrary, the only watchmaker in nature is the blind forces of physics, acted upon by natural selection. That's how you got here, class. According to Richard Dawkins and his ilk. I say it's time to revisit the argument from design, which has always been the strongest argument for the existence of God. And I say there are three discoveries we've made in the 20th and 21st centuries that all speak to a designer. The first is the universe had a beginning. We've already seen this, class. I've long since learned that there are three rules of teaching. You know this? The first rule of teaching is repetition. The second rule of teaching is repetition. The third rule of teaching is repetition. So this is the third time you will have seen this. So you're going to remember this, I hope. These are three amazing things we've discovered in this century, the last 75 years. The universe has a beginning. That's not from reading Genesis 1:1. That's from reading science. Second, everywhere you look, the earth, the solar system, the universe is fine-tuned to support life here on earth. And everybody knows it. Thirdly, even the simplest forms of life, like single-celled organisms, are full of information. I showed you this morning, there's a language in every living thing that's not only there, but it's being used at amazing speeds building stuff. In addition to that, there's machines, an untold number of molecular machines that we've just learned about in the last 20 years. That's my topic for this afternoon. So let's start with Darwin. He was, at least I think, an honest man, because in this little book, the world changed by this book right here, because it changed the worldview for many people and allowed them to be an intellectually fulfilled atheist, because they think there's a scientific answer for how you got here without God. But I'll tell you, in his book, Darwin spent a good bit of time critiquing his own theory. So read with me what he says. If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous, successive, slight modifications, my theory would absolutely break down. I'm going over this slowly, class. Darwin says if you could find any kind of an organ that could not have been formed by numerous, successive, slight modifications, Darwin's theory will only work if you can make little tiny changes that add up over time to make big changes. And that's exactly what Dawkins was saying. He said, if something is too complicated to have happened by natural causes, then just break it down into smaller steps. And if you get at that smaller step and that's still too complicated, then you just break that down into smaller steps. And once you reach a point where something could have happened by natural causes, by natural selection, that happens. Doesn't have any plan to it, it just happens. And then when that happens, you can add up that to the next one that happens. And over time, things add up so you get something really complicated. But it added up over a long period of time in small steps. You get that, class? I mean, that's the essence of Darwin's theory. And can you demonstrate, class, in nature, that you can change things naturally? Yes, you can. I mean, a classic example is dog breeds. Did you know, kids, that all the dogs we have on our earth today came from something a long time ago that wasn't like any of them? Because you can take an old Heinz 57 bunch of dogs and you can breed them to produce certain characteristics. Can you do that? Yes, you can. Darwin was a pigeon breeder. You can take just run-of-the-mill pigeons and you can breed them carefully and you can produce certain kinds of pigeons, like ones that fly off and carry stuff for you. Carrier pigeons were important back in the old days. But they were bred to do that work. And you know, don't you, there are dogs bred to do certain kinds of things? And you have everything from Great Danes, which are like about this big, to little Chihuahuas, which are like about this big. You can hold the Chihuahua in your hand. I don't recommend that for Great Danes. But I believe all of those dogs came from a common ancestry, don't you? Well, look at how much change has been wrought by natural means. Well, you say it's not natural, we bred them, I get that. But nature can also do some of that by itself. And that was part of Darwin's argument. You can change things pretty dramatically. So, are dogs kin to wolves? Well, they're pretty similar, aren't they? There's a lot of characteristics that are similar. So I don't know exactly what is the kind of dog that God created, but I know whatever kind he created had a lot of potential for variation. Is there a lot of variation in humans? Well, just look at everybody's noses. That's enough maybe to show you. We have all kinds of different variations. And what about races, folks? If you believe in creation, do you believe God created all the races separately? Do you? You think he created the black folks and the yellow folks and the red folks and all that? Or did he create one couple? So what race were they? Of course, they were white Anglo-Saxon Protestants. No, what were those first two, if that's what you believe? What were they? And where did the races come from? I'll tell you where they came from. They came from natural selection. They changed with great variability within limits. Didn't they? I mean, you're going to have to say that, creationists, unless you want to say God created every separate race. Is there any evidence of that? Anywhere? Not in the Bible. And I don't think there is in science either. I think the evidence is the races evolved from the original humans. So I believe in evolution. If you ask me, am I an evolutionist? I say yes and no, like any good politician. Depends on what you mean by that. If you mean limited change within specific limits, I mean, yes, I believe that. I think the evidence for that is very strong. But if you mean that all living things have a common ancestry in a single tree of life, from single-celled organisms to elephants, no, I don't believe that, and the evidence does not support it. Back to Darwin. If you could find a single complex organ that couldn't be made this way, my theory falls down. And I believe, folks, if Darwin were alive today and saw what we know about the cell, he'd say, I give up. My theory cannot account for that. So, I introduced this as my display. Mr. Darwin, here's my organ. Explain to me how that came about by natural selection. Lots of luck with that. It's a self-replicating, nanoscale robot. Why is it? It's self-replicating because it can reproduce itself. Class, do you know how many times in your body cells reproduce themselves every single day? I have the numbers written down because I can't remember them. All right, somebody want to take a guess? We're going to look at the process of mitosis in just a minute. That's where a cell divides and makes two exact copies of itself, mitosis. So tell me, how many times does your body mitose, if that's a verb, in one day? You want to take a guess? Somebody take me to guess. How many times do you think cells divide in half in your body in a day? In the millions. Anybody want to take another guess? That's way off. Not quite trillions. So the answer is 330 billion per day. Did you hear me? 330 billion per day. Your body is dividing cells in half and producing two identical copies of itself. So let me give you some more statistics. In a 20 to 30 year old healthy male, 154 pounds, 5'7", that's where that comes from. If you're not that, you make a little less or more. Close enough for government work. 330 billion. 86% of those are blood cells. Most of them are erythrocytes, red blood cells. And a lot of them are neutrophils, which is white blood cells. That's happening all the time. 12% of them are gastrointestinal epithelial cells. That's in your gut. 1.1% of them are skin cells. 1.1% of 330 billion class is 3.63 billion per day. That's how many skin cells you lose every day. That's 42,000 per second. Kids, take a bath. You're losing 42,000 skin cells every second. You're getting it all over the chairs. You do know, don't you, class, you're losing 42,000 skin cells every second. Did you know your skin is in layers? You ladies probably know a lot about this because you're worried about your skin. We men could care less. But your skin's growing from the inside out. And as it grows, those cells get tougher and they die, and the outside layers of your skin are a bunch of dead cells. I'm sorry. Doesn't feel like that, though, when you're hugging each other. I mean, they feel really nice. It's soft, especially ladies' skin. It's amazing. You know that's mostly dead cells, right? That's what's happening in your body constantly. And surely you studied mitosis somewhere in your career. So you know it's a complex process. I'm going to show you a little more about that today, because you don't know top side or bottom of what's happening there if you haven't studied biology recently. Well, it's self-replicating. Every cell can duplicate itself. Precisely. It's nanoscale. That means 1/billionth of a meter. That's the level you're talking about. And thirdly, it's a robot because its activities are carried out unconsciously and automatically by precision molecular machines that follow ordinary physical laws. That we've learned in the last 20 years. Lots of them. So that's our topic today. A molecular machine is a natural device on the molecular scale with few exceptions made by what class? Protein. Yes, good. That was weak, but pretty good. Able to convert chemical energy and produce linear or rotary motion. Linear motion or rotary motion. Or control biological functions. My job now is to explain to you just a little bit about molecular machines. And some of you are fading out on me. I'm watching. Here's my first one. Myoglobin. It's oxygen storage machine in your muscles. It's what keeps you going. God, give us more myoglobin. Myoglobin is made up of 153 amino acids in a row. It's a huge protein. And it works so hard in your body to keep oxygen stored in your muscles. I'm not talking about that one today, but it's the first one we discovered. I want to talk to you a little bit about kinesin and dynein walkers. They are for cargo movement, for cell division, and dyneins especially for the beating of cilia and flagella. They are working constantly. Look, if you made 330 billion cell divisions in this day, these guys were busy. Kids, if you want to have some fun, tell your mom and dad you want to go home and you want to look at the video on YouTube about Keynesian walkers. It's called A Day in the Life of a Keynesian Walker. Anybody ever seen that one? No, but you need to go see it. It's fun. It's a cartoon to tell you how hard this guy works. I want to show you a picture of a Keynesian walker. Okay? See this guy right here? This is the Keynesian walker. That's the protein. And he likes to walk on microtubules, which are also made by your cells. And he carries big, heavy stuff. I mean, this thing probably weighs 10,000 times as much as he does. And he's carrying it. He transports it. These are carrier proteins. They are critically important to lots of stuff happening in your body all the time. So we know, and if we had time class, I would teach you the chemistry of how they make their steps. We know exactly the chemistry that makes that happen. It's normal physical processes, guided by chemistry. Folks, chemistry is life, and life is chemistry. When you're talking about the material objects, life is more than that because we have a soul that was given us by God, but you can't test a soul with chemistry. But you can teach me how to walk. And we know exactly how these little guys walk. It's following chemical processes. But they do walk, class, and they carry stuff. Incredible. So, your first video today, this afternoon, we want to look at mitosis, the cell division that goes on 330 billion times every day in your life. So right now as you sit there are lots of cells dividing, and you're just sitting there graciously not even knowing about it, and going along your happy way with no consideration to how much work's going on. Shame on you. After today, you can no longer be that ignorant. All right, Troy, how are you feeling about this? He's my man. All right, Troy, we're going to now start this thing, and you're going to stop at my command. So what you're watching here, class, is an actual picture of the division of the cellular nuclear material. This is the chromosomes as they divide. Stop right there just a second. What has already happened, class, by the time we started this, was there's already been a copy made. You see, this is a copy of this. That's already happened. So I'm skipping all of that. Please forgive me. The DNA has already copied itself. Do you realize, class, how complicated that is? Because the DNA is only the information for your entire body. Everything that goes on. It copies itself without mistakes. And as I said this morning, if a mistake is made, there's built in some processes to help correct mistakes. So that's already happened. All we're doing now is starting to divide it up. So let's go ahead. What happens is the things divide out, and you watch, and you see they separate, and they're pulled apart, and then the new cell walls form by themselves, and you get two cells out of one. Stop right there just a second. Where there was one, there are now two that are identical. Not only in the nuclear material, but everything else in it. It's amazing. Now what we're going to do is we're going to start over here. Go ahead. And we're going to do it again. Only this time we're going to focus in on one particular chromosome. Right here. Stop right there. Can you see, class, that there's already two copies here? This is one. The other one's back behind it. So these are two pieces of the chromosomal material that have already been copied. we're going to go down and look at what happens to these to make them separate into two different cells. And what we're going to do is go down to the microscopic biochemical level, which you cannot see, class, so you have to have a video with an animation. And this is another, do you see the name up there at the top? This is another Drew Barry animation. It's amazing. All right, so now let's get down closer. Go ahead. These are the microtubules coming out here from both sides. And so you have two copies of this part of your DNA. And we're going to focus in now on, see this dark red? That's called the kinetochore. It's not dark red. They color it that way to highlight it. But this is where the action's taking place. Can you see that the tubules are coming out of there? There's a lot of other stuff going on here, and we're going to focus in on that. The kinetochore is what it's called. And stop right there just a second. These are the tubules that are being constructed out of, what do you think, class?
Proteins.
Thank you. And some other stuff. And it's building. And these are roadways. And the interesting thing is, as these are being built over here, way over here, they're being torn down as fast as they're being built, like any good government. The nice thing about their being torn down is if they weren't torn down, after you divide the cell, you'd be full up with a bunch of roads and you'd get all clogged up and you couldn't work. So it has to be torn down, and guess what it's done with it? It's recycled to be used again in a much more efficient way than any government recycling program we ever put together. So it's building the microtubules and it's tearing them down at the same time, but they are absolutely critical to this division process. Okay, keep going. Lots of stuff happening here. That's the microtubule attachment. Proteins are doing lots of work in here. Did you see the words there? That's a tension testing. We need a whole class on that. Did you know, class, there are
Proteins
that test for tension. And they test in such a way that when the tension is right, then you start dividing. So you've got proteins for that specific purpose, which we're not even talking about. But that's happening. Okay, so you know all this is red? He's going to start turning it green. It doesn't turn green, class. He does that because green means go. And eventually it's time to go. So let's watch. You see all this stuff happening here? There's a lot going on there. But watch. Starts turning green, you see? That means it's time to go. So you can start watching. They're going to be little carriers, we call them walkers, that start walking down that road. You see these guys? These are Dynein walkers. And they're carrying stuff. See this? Stop right there. This is a Dynein walker, class. He's got 4 legs, and he's got a bunch of stuff on him. What do you think these are, class? A bunch of proteins. Why do you think there's different colors? Because it's all different.
proteins.
Yeah, every one of them critical to the process as he carries those this way. And if you watch closely, there are kinesin walkers. There's one right there going that way. Kinesins always go this way. Dyneins go that way. Don't ask me why. That's what they do. And they're busy. And if they weren't doing this, you wouldn't have cell division. So thousands of these little guys are working while you sit there and be lazy. They're working. Keep going. Oh, I want to mention, see how he's walking? He's got 3 legs, doesn't he? Right here. Did you see the kinesin walkers? There's one right there. See him? Stop. How about these blue guys? What do you think they are?
Protein.
Exactly. And they are very important because they keep these microtubules in place and they guard that whole system. And so you got walkers going this way, walkers going that way, lots of activity taking place. And by the way, this is way too slow. It's slowed down so you can see it. Keep going. I love it. Don't you love the way they walk? Stop right there. Now we've gone back out to the larger vision, but I want to tell you again, we know exactly how they do this. And they do it rapidly. And there's a lot of chemistry involved there. Now we're back to the big picture. When the time is right, watch what happens. The microtubules with their entourage says, let's move it. And there it goes. But you know, class, there's a bunch going on you don't even see. A bunch of machines doing amazing things. 330 billion times per day. Isn't it wonderful what nature did, one step at a time over millions of years? Natural selection's amazing. Folks, there is no way natural selection did that. That was designed. And we, can I just say this? We don't know doodly squat about all that. What I'm telling you is probably the tip of the iceberg, which we have yet to uncover. Let's talk a moment about cilia. Kids, do you know what a cilia is? It sounds kind of silly, doesn't it? Any of you kids know what a cilia is? It's a hair. And did you know you have lots of them? Not just these. You have some in your nose. I'm sorry, that's kind of not a thing to talk about. But you have other, much more important hairs in other parts of your body, some of which I will refuse to discuss. There's some very important hairs right there. Right here. Here's your mouth. Here's your bronchial tube. And it enters into the lung. There's lots of hairs right there. Why, class? Why are these hairs right there? Tell me. To filter stuff that has no business in your lungs. That's why students, children, don't smoke. That's the stupidest thing you can do. Because you're messing up these cilia, and you don't want to mess with cilia. They're critically important to your being alive. So you have thousands of them right in here. What you don't know, class, is every one of those hairs has to be built by your body. And it is built from the skin level up, or wherever it's coming from the bottom up. And there's little kinesin walkers that take the stuff out this way. There's little dynein walkers that carry it back this way as the job of building that cilia takes place by the thousands per day. So that's another thing that's going on all the time. Just to build hairs. And your life depends on it. And if I had time to explain all that process to you, it would take me the next 2 days. But the cilia are incredible. I don't have time to tell you all of that. I want to focus in on one motor, one machine. It's called the bacterial flagellum. It's a rotary filament. It's like this. But something has to drive that motor. So there's a motor connected to that filament that rotates it. That's what I want to show you about. But before I show you that, I want to share with you some statistics. This little bacterial flagellum and its motor are located in a little one-celled organism that has a tail. And the tail is the flagellum. Everybody lift your thumb up like this and turn it sideways. You see the width of your thumbnail across this way? That's not very wide, is it? You can stack 1,000 of these bacteria across that width. Did I get myself clear? That's how tiny these guys are. One of the big ones we want to talk about is Escherichia coli. otherwise known as E. coli. What do you know about E. coli bacteria? You don't want it. Because why? It'll make you sick, and it can exactly kill you. And if it gets in food, all the food has to be destroyed. But did you know, class, that there's a strain of E. coli that is absolutely critical to your life? Did you know your intestine is full of it? I'm sorry, kids, I have to talk about your intestine. You have little bacteria swimming around inside of there all the time. Sorry, but you do. And if you didn't, you'd be dead. I'm sorry. But you have millions of E. coli in your gut. Are you with me, everybody? So we're going to talk about E. coli, and the E. coli's little flagellum is driven by a motor the likes of which no human has ever produced. Before I show it to you, here are the statistics. First of all, Howard Berg, Harvard biophysicist, says of the bacterial flagellum, it's the most efficient machine in the universe. The first time I talked to this, my Marilyn was in the audience, and she said, "You lost me after the first two sentences." She said, "Make sure everybody understands exactly what you're talking about so they stay with you." So can I get your attention? I'm talking about your intestines. Are you with me? There are millions of these little guys swimming in your intestines right now, and if they didn't, you'd be dead. Did I make that clear enough? So is this real stuff we're talking about? It's life and death is what we're talking about, and these guys can swim. And it's because of their flagellum. And you tell me when I get finished with this, that motor and that flagellum came about by natural selection, acting on natural variation over millions of years. And I'll tell you, I have a bridge to sell you down in the Everglades, which doesn't exist, by the way. The bridge doesn't. From Harvard biophysicists, here's some facts. This motor rotates from 6,000 to 17,000 revolutions per minute. I presented this to a guy who was into car racing. He said the best racing engines go about 15,000 revolutions per minute in the best racing cars we have. This little guy rotates up to 17,000 in a little one-celled organism. The filament, which is connected to it and rotates, goes anywhere from 200 to 1,000 revolutions per minute, or 17 times per second. Watch me, see if I can do this. In 1 second, it goes. Can you do 17 times in 1 second? I can't either. That's how fast this little thing goes. But here's the amazing thing, class. If it's going clockwise like this, at that speed, in an instant, it can go reverse clockwise at the same speed. And it'll take that little bacterium who's swimming this way, and it'll turn him this way, just by turning the rotor the opposite direction. There is no thing we've ever created that can do anything close to that. And it's in the little one-celled organism in your gut. Doing important stuff now. So let's see if I got any more facts here. Oh yeah, here's another one. This little guy can swim faster than a cheetah runs. Now remember, this is on his cell, and we're talking about nanometers. But that little organism can go 60 lengths in 1 second, 60 of its own lengths. Well, that's only a few nanometers, but look, for him, that's whipping along. A cheetah can go 25 body lengths in a second. You see what I'm saying? A cheetah can run 25 of its body lengths in a second. This little guy can go 60 of his body lengths in a second. And here's the other astounding thing. he uses almost no energy to do it. If you had time, I'd explain to you how it uses an acid imbalance in its motor to drive the motor that uses almost no energy. You should have fallen out of your chair, class. Why aren't you falling? This is incredible. That's what we're dealing with here. And now I have a video made by a Japanese company, not by Mr. Berry, but by a Japanese company, that shows you about the flagellum. That's going to conclude my lecture today. So I'm going to get over here. All right, Troy, let's go. This is an actual picture of the bacteria swimming around. These guys are pretty good. But what you're going to see next is the E. coli. You watch. There's the E. coli. Stop. Here's one of those. Do you see? He's got lots of flagella. An E. coli can have anywhere from 4 to 10 flagella. And they wrap all around each other, so when they do this, I mean, they drive that thing. It's amazing. He's one of the best swimmers there is. So these guys are getting around. They're very busy. You understand what I said, right? In your intestines right now. So let's watch. Go ahead. Now you can't see them very well, so what's going to do is they're going to make an animation of the bacteria. This is the bacteria itself. This is the flagellum, and stop right there. You may have noticed there were 4 of these attached. Each one of them is attached in a particular way to the cell wall of the bacteria. So we're going to look at the parts of these now. And by the way, I forgot, because some of you are going to sleep. Everybody stand up. Shake yourself. I don't want to lose you here in this last piece. If I would have you do calisthenics, if I thought it was appropriate. How about jumping jacks? Get your blood flowing, kids. You're not allowed to sleep. Okay, good. And nobody left. That's good. Have a seat. I hope that helps you, because it sure would me if I were sitting down. All right, I want your rapt attention here. I'm going to tell you about this bacterial flagellum. Let's go on, Troy. And when the words come up here in a minute, we're going to stop. Right. Where are the words? No, there's one, and there's another. Stop right there. All right. Here's the connection, class, between the filament and the cell wall. The filament, can you see that's a different color? That's because it's a different... Can you be a little louder on me? It's white because it's a different... Protein. Thank you. The protein is filamin. You don't have to say that. But may I tell you about filament just briefly? It's perfect for a filament because it's flexible, it's strong, and you can wrap it around it, and it's almost indestructible. It's amazing stuff. But it's a different protein than any of the rest of them we're going to talk about. All right. Do you see these dark ones? There's a kind of real dark one and another dark one. That's different. Protein. Are you awake? All right. That is different. Protein. Yes. And then you connect up to the hook here, which is a different. Protein. Exactly. Kids, listen to me. This hook has to be exactly 54 nanometers long. Exactly. If it isn't, it doesn't work. And you don't want a hook that doesn't work, because then the bacteria dies. So how is it that this little guy builds a hook exactly 54 nanometers long? It's because of protein that have exactly the right number in the right order and the right placement until the right number have been added, and then they stop. That's the hook. Critically important. Let's keep going. Now we go inside here, and it connects through the cell wall. Stop. You see these bushings here? that help keep it alive. If they didn't have bushings, you'd have an open hole there, and the thing would be dead in a matter of microseconds. But those bushings are there for a purpose. Darwin said, if you can show me anything that could not possibly have been formed by small successive steps over a long period of time, well, here's one. And there are millions of others. All right, so we go on down. We're looking at the various parts. Keep going now. There's some words that will come up here in a minute. What you're watching is the motor. And it has a rod going right down the middle. Stop right there. You got a bunch of rings. You see this? Different color rings. That means they're different what class? Yes, all these are different proteins, specific for their specific purpose and in a specific order in critically important rings. And here is the layers of the skin of the bacteria. It has 1, 2, 3 layers. That little tiny thing, 1,000 of which you could stack across that thumb width, has skin that's 3 layers thick. And every layer is critically important. You see this peptidoglycan layer, the middle one? The stator here, you see these connecting rods? Connects to the stator. Let's go on and see if it tells us about that. Hang on. Is he going to tell me the stator here next, I hope? Yeah, stop right there. The stator. Class, if something is rotating at a rate of 17,000 times per minute, do you think it might vibrate a little bit? I mean, it would shake the thing to death in no time. That's why you have a stator. Every motor built by a human that's like this has a stator. So does this. And these rods go all the way around, and they're anchored in the peptidoglycan layer of the skin of that little organism. So it keeps that motor from vibrating. Does that look like a plan to you, class? Let's keep going. And then down here you have the basal body. And I'm leaving out 68 things, you know that, in the process here. So this is the motor. Now stop right there. Here's what you have to understand. Maybe if you don't remember anything else from today, I want you to remember this. This motor is not reproduced when the cell divides. You know bacteria divide and make more bacteria. When that bacterium divides, it does not produce the flagellum when it divides. It has to be built once it divides. You with me? It takes about 20 minutes to build a new flagellum in these little guys. And we know exactly how they do it. You might be asking me, why have we studied E. coli so much? Because it's a killer and it's a savior. It's a savior and a killer at the same time. We want to know everything there is to know about it because it helps keep you and me alive and not dead. So they've studied this guy more than any other creature on the universe. That's why we know so much about him. And here's what we've learned. That little creature, once it divides, builds that flagellum from the bottom up. Every piece of it. We know exactly how he does it and the order in which it happens. And it's done without a straw boss telling everybody what to do. It's guided by the DNA and the proteins who do all the work. And there's a bunch of different proteins here. So what you're going to watch here is the building of a flagellum that happens automatically every time the bacteria divides. So may I tell you the conclusion of that? Not only do you have one of the most remarkable motors in the universe, as the Berg guy from Harvard said, but you have the mechanism to build it. So it's like not only do you have a Ford motor, but only way better, you have the factory that builds the Ford motor also. And it builds it in amazing speed with no mistakes. I want you stupefied. This is not possible. And people sit around, intelligent people, and talk about, wonder how that evolved. I say, what is wrong with you? What does God need to do? Take down and shake you? He's already done it. All right. I hope I made that clear now. Let's watch them build. Here we go. The first thing that is constructed is this. That's exactly 26 copies of the fly F protein. That's constructed just like that. And then the fly G protein is added, exactly 26 copies of that different protein. And then the fly M and fly N proteins are added on the bottom. In this order, class, stop just a second. It has to be in that order for all kinds of reasons, but the body does it precisely in that order with the right number of everything that's put together in the right order. Some of you guys work in a factory or in a production place where you know how many things have to be done. This is all done automatically by the little bacteria itself. And tell me that wasn't designed by a master engineer. Keep going. The next thing that's built is the rod coming up through the middle. Do you see it's white? That's another different protein. So as the rod comes up through the middle, it has a cap on it. Why does it have that cap? Well, stop right there, because first you got to build a stator. That's the next thing, and we're not even talking about that. That's another piece, but it's in this order. And now this cap is going to drill a hole through the rest of the cell wall. Do you think that's a healthy thing for a little E. coli? Not only kill it. You drill a hole through the wall, it'll kill it in no time. But what happens is exactly at the right time, in the right order, these bushings are added while the hole is being drilled. Go. There are the rod proteins and the rod cap. It's driving the hole. And immediately you get these shells, these rings, the L and the P rings. Stop right there just a second. I got to tell you this. The L ring would never form if you didn't have a chaperone. That's what they call it. It's another
protein.
Good. And the job of that protein is to chaperone the specific protein for the L ring up to the right place and put it there in order. And if you didn't have a chaperone, you'd have no L ring. And if you have no L ring, what do you think? You have a dead E. coli. So included in this complex process is a chaperone protein to make the L ring form. Keep going. Now, that other cap is, stop. That other cap has been released and recycled, by the way. You make now a hook cap. That's specifically to build the hook. You remember I told you it has to be exactly 54 nanometers long. There's a hook a cap that guides that process and puts these in exact right order and builds the hook. Watch. Stop just a second. Do you see that it's adding one piece at a time and putting them in place right where they belong? Like this. Go. And when that hook is done, you dismiss the hook cap and you add these extra two sets of proteins, the rod junction proteins, and then down up through the middle, coming here, you get a new cap. This is called the filament cap. Stop right there just a second. It has 1, 2, 3, 4, 5 little pieces and 5 legs. Kids, watch me. There's a name for this. We call her Twinkle Toes, because she dances around to build that filament. So watch me. It's like this. Every time she lifts up her leg, that leaves a hole for a specific filament to come through there and be added. Then she sticks that leg down and it lifts this one up, and another protein comes in the other place. And she rotates around, and that's how that thing is built. The Twinkle Toes is incredible. And anybody want to take a guess? How many pieces of filament protein it takes to make one long filament? Take a guess. I mean, you're pure guessing, right? 32,000. Let's watch it. All right, twinkle toes. Can you see as she lifts her step, they come in? And they're in a certain specific order. That's way too slow. Shape up. Move it. No, that's more like it. Go. That filament is about 10 times as long as the little bacteria. And it's just constructed it. In this case, he did 3 others at the same time. So you have a factory that's building multiple flagella at the same time in the process I just showed you. I know it reaches a point where you're just numb. I want you walking out of here stunned. And we know how this works now. There are no excuses, class. If Darwin were standing here today, I firmly believe he'd say, I give up. This is not a Darwinian process. Darwin's theory works only within limits. It accounts for the various breeds of dogs. You can have limited change, but it does not account for something like this and the complexity of nature all around us. No, class. God has overwhelmed us with evidence of design. And I don't care how you want to break that down. It didn't happen by natural selection, acting on natural variation, by Darwinian evolution. Because it's a multiple protein process. Each protein is complex and suited for its function. Each machine is highly sophisticated, intricate, dynamic, and elegant. These machines are not only there, they are spontaneously self-assembled in the cell. And there are thousands of them in every living cell. So I ask you, class, which is the more reasonable explanation for molecular machines? Here's what Dawkins would tell you. They were produced by the blind forces of physics through numerous successive slight modifications produced by random variation acted upon by natural selection. It's the modern day fairy tale. Or were they designed by a supremely intelligent grand designer? Now, I know that scientists will say that's just a cop-out. You're not explaining anything. Oh, yes, I am. We know how things are designed. We know how to do it. The evidence is overwhelming. Somebody designed this. It's not based on ignorance. It's based on what we know more than ever. And must be supremely intelligent. Because we know what designers can do. We do it all the time. I say to you, class, you've never lived in a time when there was more reason to believe in a God than you do right now. And you've been a great class. Thank you for your careful attention. I pray to God, my goal is to stupefy people to the point that they love God so much because of who He is and His amazing power. And it makes me want to take people to His other book in which Jesus said, if you believe in God, believe also in me. Don't let your heart be troubled. I go to prepare a place for you. And if I go to prepare a place for you, I will come again and receive you to myself. So what people need to hear today is not only there's evidence for God everywhere. You need to be a God believer. And then you need to learn about Jesus because He's the Savior of the world. And He's your only hope. And you can come back to be with Him forever one day and live a life of complete and utter purpose in this world as you prepare for the world to come. Is it reasonable to believe in God in this scientific age? I say, amen.